Hello, everyone. Today, my team and I will be presenting our project titled Ozone-Driven Antimicrobial Sterilization for Enhanced In Vitro Culture Sustainability. We'll explore how ozone offers a cleaner, safer, and more sustainable alternative for seed sterilization in tissue culture. Seed sterilization is a crucial step in successful in vitro culture. However, many conventional chemical methods are harsh, toxic, and can reduce seed viability. This project explores ozone as a cleaner and more innovative alternative, offering strong antimicrobial power while supporting sustainable tissue culture practices. Traditional sterilants like ethanol and sodium hypochlorite are widely used, but they come with several issues: seed coat injury, tissue browning, reduced germination, and inconsistent contamination control. Ozone, on the other hand, is a powerful oxidizing molecule with high antimicrobial activity and a short natural half-life, breaking down safely into oxygen. Because of this, it has strong potential as a low-toxicity disinfectant for Solanaceae seeds, such as tomato, cherry tomato, and chili. Chemical sterilants often cause seed injury, inconsistent contamination control, and lowered germination in Solanaceae species. This project aims to determine the effective ozone exposure time that reduces contamination, identifies the remaining microbial types, and enhances seed germination across different species. Our research objectives are to compare ozone and the bleach ethanol method for contamination control, to identify bacterial or fungal contamination that remains after ozone treatment, to determine the optimal ozone exposure time for seed germination. To bring this idea to life, we designed a hands-on experiment that carefully follows each step from seed to culture. We began by preparing seeds from 4 Solanaceae species: bird's eye chili, red chili pepper, common tomato, and cherry tomato, all sourced from the Jengka, Pahang area. Seeds were extracted and washed under sterile conditions. Murashige and Skug basil medium was prepared and poured into 36 sterile petri dishes. For treatment, 18 seeds were used per method, with 6 seeds per plate. The control group followed the standard sterilization method using 70% ethanol, followed by 50% sodium hypochlorite with a drop of tween 2.0 and several sterile rinses. For the ozonation group, a 500 milligram per hour commercial ozone generator was used. Seeds were submerged in ozonated water for either 4 minutes or 8 minutes before inoculation. Data were collected over 2 weeks, focusing on the frequency of contamination, the frequency of germination, and the frequency of necrosis. After 2 weeks of growth, the outcomes became crystal clear and the seeds had a lot to show us. The first slide showcases 5 examples of outcomes observed during the study: healthy seed germination, bacterial contamination, fungal contamination, combined fungal and bacterial contamination, and seed necrosis. The next slide presents 2 graphs. One showing the frequency of germination across treatments, and the second showing the frequency of bacterial contamination. These visuals help illustrate how different ozone exposure times compare with traditional sterilization methods. Ozone sterilization offers several environmental benefits. It reduces the use of hazardous chemicals and leaves behind no toxic residues. It lowers ecological toxicity, supports green laboratory practices, and requires very little energy to produce. Overall, ozone provides a safer and more sustainable approach for both researchers and the environment. In conclusion, ozone-driven antimicrobial sterilization presents a promising and sustainable alternative for contamination control in vitro culture. It enhances seed health while reducing dependence on chemical sterilants, paving the way for greener and more innovative tissue culture practices.